Assalamu alaikum, students. So this is the final topic of renal physiology. Uh, when I say final topic, I mean final major topic. Of course, there will be some topics which will be left and will be addressed accordingly in short videos later on. So acid-base physiology or acid-base balance or acid-base disorders, it has many names, but I'll stick to the term acid-base physiology, which encapsulates all of these concepts. And in some books, it's a whole chapter, as is the case in Guyton. So definitely, it's not only from the physiological point of view, it's a very important phenomena, but also it links directly into clinical practice. And often what is seen is if your physiology about these matters is weak, it has direct effects on your practice of medicine. So with that, let's move on and look at part one of this big unit, which will be dealing with basics of acid, base, and buffers. We'll be talking about what are the lines of defenses against imbalances in acids and bases in the body, and we'll finish this particular video by looking at what exact role does the kidney play in acid-base balance. So back to the basics, you know all of this essentials of pH. We know that hydrogen ion concentration is a very, very small numerical number, as you can see right here. It's actually in nano equivalents per liter, which is very minuscule. And for ease, what we do is we convert the hydrogen ion concentration, the actual concentration, into an algorithm, a log. We call it pH. And there we have a manageable figure of 7.4, which obviously is very commonly known. It is the average, the middle of the range of the pH. pH actually has a range. Okay, we will talk about that. Okay. So, by the way, this is the range of the pH. So, when you say 7.4, it does not mean that 7.38 or 39 is not normal. It is. The range is, the normal range is in the human fluids, it's 7.37 to 7.42. Okay. Anyhow, let's start with the graph. So basically, it's a graph between the y-axis is hydrogen ion concentration and its expression in pH. This is right in the middle, the normal pH, which is 7.4. And what is interesting is you see the graph downwards, which corresponds to an increasing pH. We call it alkalemia or alkalosis, or if you go above, i.e. you decrease the pH, it's basically acidemia or acidosis. Okay. The shape of the graph, if you really concentrate on, it's interesting. If you if you drop this pH and just follow the graph from 7.4 to 7.6, you have a change of hydrogen ions approximately equal to 15 equivalents per liter. However, if you do the same amount of change and decrease the pH from 7.4 to 7.2, the amount of hydrogen ions that are involved here is 23 milliequivalents per liter. So larger change in hydrogen ion happens or occurs in the acidic range than in the alkaline range. This is an important peculiarity in the way hydrogen ions function in an acidic pH as compared to an alkaline range of pH. Okay, as I mentioned before, this is the normal range, 7.37 to 7.42. Anything less than 7.37 is called acidosis. Anything more than 7.42 is alkalosis. And generally, the extremes of compatibility with pH is 6.8 to 8. Of course, these are not comfortable pH values, but still somebody can survive to some extent during this wide and wild swings in the pH. Okay, So very basic about acid base. What is an acid? Acid is a substance that can release or donate hydrogen ions. A base is a substance which can accept it. When they combine, they can be dissociated into hydrogen ions. An acid, when it's added to the water, it can be dissociated into hydrogen ion and a conjugate base. Acid and alkali in the body is basically, where does it all come from? So diet is basically a mixture of acid and alkali. Then you have your, so whatever you have in terms of food provides acid and also provides alkali. It's a source of that. Cellular metabolism also yields acid and alkali. And feces basically is the source or the route via which you excrete most almost all of the alkali that is either acquired by the diet or produced in the metabolism. Okay, so that's not the problem. The problem is acid. The acid that you make that needs to be sorted out, and the whole acid-base balance basically almost in the daily mundane basis looks at how acid is to be sorted out. I'm not talking about the disorders, I'm talking about the everyday life. So every day you produce a certain amount of acid which will call net acid, and it needs to be excreted. So NAE, net acid excretion, is that one process that the body needs to invest in basically. Okay. Right. So where does what comes from? Basically, this is a very important slide. So dietary stuffs is CHO is basically carbohydrate. Please remember, this is not a standard abbreviation. This is just, I just wrote it to decrease the word count of this slide, because then it becomes very crowded. So carbohydrate and fats, basically, when they are properly consumed, properly metabolized, they release or they produce carbon dioxide and water. Carbon dioxide, as you may remember from your chemistry classes, is called a volatile acid because you can, well, literally exhale it. Okay, so no problems there. The respiratory system is used here to remove the volatile acid that is formed from carbohydrates and fats. And water is not a problem. Now, other food items, that is mainly protein. These are basically, these are three main dietary forms, isn't it? Carbohydrate, fats, and proteins. The proteins are different in the sense that they produce non-volatile or fixed acid, like HCl or sulfuric acid. So it's this non-volatile means you can't exhale it. The respiratory system has nothing to do with this. This needs to be sorted out by something else. And we'll obviously, we know what that something else is. It's the kidney, okay? So non-volatile acids are sorted out by kidney. Bicarbonate that is formed, that is also formed in metabolism, plays a part in neutralizing these fixed acids that are formed. However, the main source of addressing these strong acids, as I said, is the kidney. The kidney needs to sort this out. And this is the net acid secretion basically, which is between 50 to 100 milliequivalents per day for a person who eats a slightly more protein-rich diet, which is the case these days in most part of the world, unless the person concerned that who we are considering is a vegetarian or a vegan. Generally speaking, non-vegan and non-vegetarian people consume more proteins than other food groups, and hence the net acid secretion load is about 50 to 100 milliequivalents per day. Okay. Now there are certain abnormalities of net acid secretion, and these are the ones. So what is common between diabetes mellitus, which is basically the lack of insulin? We're talking about type 1, hypoxia, which by now you know is decrease in oxygen concentration, such that the body's tissues are deficient in oxygen, and cardiac failure, where there is a decrease in cardiac output. So normally we have normal insulin secretion, where all the carbohydrates, oops, sorry, all the carbohydrates, when you're normal, you're not diabetic, all carbohydrates and fats are basically metabolized to produce carbon dioxide and water. As I said, carbon dioxide is exhaled, not a problem. However, when a person develops diabetes mellitus, i.e., insulin is lacking, what you then have is carbohydrates now will be metabolized not into carbon dioxide and water, rather, they will be metabolized to keto acids. Now, keto acids are another form of acids which you cannot exhale, so you have a problem now. Okay, so these keto acids, an example of it is beta hydroxybutyric acid, and these are the acids basically which add to the acidosis of diabetes. We call it diabetic ketoacidosis. Okay, so diabetes is accompanied by a specific type of acidosis called diabetic ketoacidosis, DKA. Okay, you will hear a lot of this in your medicine ward, unfortunately. So basically, in diabetes mellitus, you have this sort of problem, this sort of abnormality, which adds to your net acid load. So this chap will produce this anyway because this is the normal load anyway. But in addition, this person will also be producing keto acids, which will add to the net acid load. All right. So that's one. What about hypoxia? So hypoxia, basically, there is an aerobic respiration, then there is anaerobic respiration. Aerobic respiration requires oxygen. Anaerobic respiration is switched into when you have lack of oxygen. And this is what happens in hypoxia. There is a lack of oxygen. The tissues have no choice but to switch into anaerobic respiration, which has this side effect of producing other fixed acids such as lactic acid and this, that, the other, again, which are organic acids. They cannot be exhaled out. They're not volatile. And that can add to the net acid load. You may be familiar with vigorous exercise uh, causing pain in the muscles, especially if you are not accustomed to that level of exercise and you suddenly end up doing it. Or commonly called as cramps. These are basically due to accumulation of lactic acid in the muscles. All right, and this is an acid, and hence it needs to be sorted out. Okay, what about cardiac failure? So cardiac failure again is low cardiac output. Basically leads to a decrease in perfusion of the tissues, and this again has the same effect. The tissue will go into anaerobic respiration and produce organic acids. Now, what is the solution for this? So in diabetes mellitus, give insulin. When insulin is present in the system, carbohydrates naturally then switch back into conversion into carbon dioxide and water. Problem solved. In hypoxia, restore the oxygen. Cardiac failure, improve the cardiac output, so that tissue perfusion improves. When tissue perfusion improves, oxygen is there, oxygen lack is addressed, and hence the body switches into aerobic respiration, and that sorts these things out. Okay. Now, buffers. As you may remember, buffers are agents or chemicals which minimize change in pH when in a solution you add acid or base. So these are agents. They don't prevent anything. They basically minimize the damage if you put in an acid or a base to any solution, be it ECF. So you can now visualize that cells continuously make acids and all sorts of base, and then in when you eat, you have all sorts of acids and base that you end up eating. So all of this has potential to change the pH of the ECF, which is not recommended and is fought against at all costs. And one of the mainstays of that fight is basically to buffer it chemically with a buffer, where weak acids and either its conjugate base is used, or a weak base and its conjugate acid is used. So actual examples of buffer systems in the body are divided basically into three, as I can put it here: extracellular fluid, intracellular fluid, and bone. In this, basically, you cannot afford to forget this stuff here, right here, and of course this here. Okay, bone is covered in endocrinology, but this is the important stuff. So extracellular fluid, the most abundant buffer system is this bicarbonate carbon dioxide buffer system. And you can see that the carbonic acid, sorry, carbon dioxide, when it reacts with water, it forms, irreversibly it forms carbonic acid, which then dissociates into hydrogen and bicarbonate. So in reverse, when you produce hydrogen ions, if you have bicarbonate ions handy, you immediately titrate them or combine them with your newly formed hydrogen ion and form carbonic acid, which is a weak acid, mind you, and which easily gets dissociated into carbon dioxide and water. Carbonic anhydrase is the enzyme which does the needful, and there you have it. Your hydrogen is now packed inside water and can be of no consequence to pH. Okay, so this is the most abundantly present buffer system in the body ECF. The other one is inorganic phosphate, and here you can see that if you have your hydrogen which is being produced, you can buffer it with hydrogen phosphate anion, and basically this will form H2PO4, which is weak and can be dealt with by the body appropriately. Then you have plasma proteins which have buffering action. They combine hydrogen ion, and what you are seeing basically is the body does not tolerate hydrogen ions in the free form, and has to be these have to be packaged here with bicarbonate, here with HPO4, and here directly with plasma proteins. Okay. And then intracellular fluid has cell proteins. You have been studying this since your start of the MBBS, that the cells in themselves have lots of protein molecules inside them, and one of the functions of them is basically to act as a buffer. RBCs are equipped with hemoglobin, which along with oxygen carriage also has a buffering action. Here you see hydrogen being buffered by hemoglobin. Then you have organic phosphates. Again, basically hydrogen is being buffered here. Then you have the same carbon dioxide bicarbonate system, which is present also in the intracellular fluids. And bones, you have again phosphates and carbonates doing the job. So in kidney, basically, you'll be dealing with this buffer system, the bicarbonate carbon dioxide buffer system, the inorganic phosphate buffer system, mainly, and there is an ammonium buffer system, which is not shown here, and it's most prolific inside the kidney. We'll be talking about that in a while. Okay, so this is the lines of defense concept. I like this line of defense concept. It's similar to what you studied in blood physiology, lines of defenses against any pathogen entering the body, you know, the lines of defenses of inflammation. So similarly, we have lines of defense against any change in pH, and it's nicely made here. It's nicely depicted. Food intake, digestion, absorption, basically cell metabolism inside the cell, having all these reactions going on, produces three things. One is these fixed ions, your sulfates and your chlorides, which are strong and will give rise to a strong acid. They basically are produced inside the cell. You have carbon dioxide, which is being produced inside the cell, and you have free hydrogen that is being produced inside the cell. We'll start from the free hydrogen because this has the potential to really drastically affect pH, drastically. So as soon as they are made, hydrogen ions are basically combined with intracellular proteins. If some of them leave the cell, there is chemical buffering outside available. The carbon dioxide, the carbonic acid buffer system, basically, which is abundant in the ECF, as I mentioned, basically titrates hydrogen, and in this process basically consumes the bicarbonate. And this is an important point here, which I want you to log away as an important point, that every time you excrete a hydrogen out of the cell into the ECF, a bicarbonate ion is there, which combines with the hydrogen and starts that carbonic acid and then the whole carbon dioxide water reaction. But what I want your attention on is this bicarbonate ion, which is consumed by every hydrogen ion that is secreted. So this needs to be replenished. Please remember this. Bicarbonate ion, which is available in the ECF, gets used up on a daily basis by production of free hydrogen, and that this bicarbonate, this needs to be replenished. This is a very important point. The carbon dioxide that is produced is not a huge problem in the sense that it's not fixed and it doesn't consume much in the sense which needs to be replenished afterwards. Carbon dioxide basically is exhalable and you exhale it through the respiratory system. Okay, so the first line of defense is chemical buffering, which we discussed, intracellular and extracellular. The second is respiratory, where carbon dioxide you exhale. The third is of these ions, which may form, if not sorted out, may form proper hard fixed acids, and they basically are sorted out by the kidney in various forms and excreted away from the body in the urine. So the first two, if you want to create a timeline, is rapid acting, while the renal is delayed onset as usual, but it brings the most notorious ions to their fate. So it does come in late, but it does the most difficult job, as is the case with other processes like blood pressure and ECF volume and so on. Okay. So with this, we have done the basics. Again, there is all books are available. Guyton, especially, you can have a read of the basics where it talks about buffers in much more detail than I have. I've just given you an idea. You can always read up and increase the depth of your knowledge by following a standard textbook. Okay, now we'll talk about renal, because this is, i.e., this is part of the whole unit. We are discussing kidney. Uh, Mainly, kidney's acid-base balance. There are two roles that it basically rotates around. And the number one thing you might find this a bit awkward is that excretion of hydrogen I have put at number two, and actually at point number one I have put reabsorption of bicarbonate. Uh, but this order has some thinking behind it, because reabsorption of bicarbonate is uh, extremely important. Why is it important? Well, we just mentioned it. The most abundant buffer system is your carbonic acid, that carbon dioxide, carbonic acid buffer system. And it requires bicarbonate. If you remember, let me just go to 2 steps back. Remember this bicarbonate? What if I remove this bicarbonate? It's not here. And now you add hydrogen. What will happen? Nothing will happen. Okay, the buffer system won't work. It needs bicarbonate. When hydrogen comes, it's the bicarbonate that gives up its life, so to say, and make carbonic acid, and then it dissociates into carbon dioxide and water. You exhale carbon dioxide and water, we welcome anyway. So really, it's this chap here which is crucial, and you lose it every time you have a hydrogen hitting the ECF, right? So reabsorption of bicarbonate. So you freely filter bicarbonate. Lots of bicarbonate gets filtered at the glomerulus. You need to catch it. You need to reabsorb it. Okay. So one of the most prominent functions of your renal tubule is to reclaim this filtered bicarbonate, as we'll see in the subsequent slide. So very, very, very important function of the kidney in acid-base disorder is to recover bicarbonate as the number one function, quantitative function. The second is, of course, excretion of hydrogen as a titrated acid with the urinary phosphate system. We'll talk about this. Number two, as an ammonium ion, we package the hydrogen together with ammonia, NH3, to form ammonium, which is a weak acid, and we excrete it. And then we can secrete some of the hydrogen per se as such. Uh, uh, Basically, this per se hydrogen, most of it is used to trap filtered bicarbonate. Let me just clarify this in a bit. First, let me just revise this whole thing. Excretion of hydrogen is coupled with phosphate. So you titrate it and excrete it as a weak acid in the urine, number one. Number two, you combine it with ammonia gas, NH3. You know, the smell of urine is of ammonia. Remember this by that. So NH3 combines with hydrogen to form NH4, which is a weak acid, and so it can be easily excreted in urine. So that's that. Number three is secretion of hydrogen per se in free form. Okay, and in this free form, out of this free form, most of the hydrogen is used to trap bicarbonate. You can imagine that you are fishing and you are using a rod with a bait at the end to attract the fish. Okay, so you basically secrete hydrogen inside the tubule to trap the fish. Bicarbonate is the fish. The filtered bicarbonate. The filtered bicarbonate needs to be trapped because it can easily go through and be lost. Your precious currency can be lost in urine. You don't want that. So you secrete hydrogen and you take even benefit from this by trapping bicarbonate. And the little amount of hydrogen which per se is lost in the free form basically gives the urine its pH value, which is, of course, more acidic than ECF fluids. Okay, as you must have noticed in the initial slides that I showed of urinalysis, the analysis of urine as a fluid, and you must have must have noticed that the pH values are lesser than 7.4, way lesser than 7.4. Okay, then when we've done this one bit is left here. These two, the titration of hydrogen against phosphate and the combination of hydrogen with ammonia to make ammonium, they form new bicarbonate. They form new bicarbonate. Basically, this new bicarbonate is formed, created. This bicarbonate is filtered. So it is coming from your plasma. This is filtered bicarbonate. This bicarbonate, that's why I've given it a different color. It's formed by the kidney. So this is a very important distinction. This is one of those pet viva questions. What is the difference between the newly formed bicarbonate, who forms it, etc., the difference between new formed bicarbonate and filtered bicarbonate. It's a very important distinction. We'll talk a bit more about that. And just a footnote to this particular slide is a quick note of how much bicarbonate is filtered and how much of it is reabsorbed. I'll share with you some statistics. So basically GFR per day, as you remember, is 180 liters a day. Yes. The plasma bicarbonate is 24 milliequivalents per liter. You multiply this and you get a big figure, 4,320 milliequivalents of bicarbonate a day is the filtered load of bicarbonate. Once again, GFR 180 liters per day, plasma bicarbonate 24 milliequivalents per liter. You multiply this, you get the filtered load of bicarbonate, 4,320 milliequivalents per day, right? Guess how much you excrete eventually of bicarbonate on a daily basis? It's hardly only 2 milliequivalents per day. So 99.9% of filtered bicarbonate you capture. So now this is the context of uh, this reabsorption, of which hydrogen secretion is one of the mainstays. So hydrogen is secreted, it traps, it combines with, or it helps you reabsorb your bicarbonate. And at the PCT, this is one of the mainstays of bicarbonate reabsorption. Remember, 99.9% of bicarbonate, filtered bicarbonate, needs to be reabsorbed. Okay, so what happens in the kidney? Let's go in a bit more detail. We'll go one by one. So number one point was bicarbonate reabsorption, right? So this is a PCT cell. PCT is the site of the most bicarbonate reabsorption. However, it does get reabsorbed at the loop of Henle, DCT, and CD. 99, as I mentioned, 99% of filtered bicarbonate is normally should be reabsorbed. Now, what happens? What's going on? This is the PCT. You have carbon dioxide and water freely available. It gets combined in the presence of carbonic anhydrase, which is the enzyme facilitating the forward reaction, H2CO3, carbonic acid. The carbonic acid immediately decomposes into hydrogen and bicarbonate. Okay, this hydrogen, just follow this highlighter now. This hydrogen now basically is secreted as part of a antiport mechanism with sodium. And if you remember, I mentioned this when I was discussing sodium reabsorption at the PCT under sodium handling by the nephron. Okay, if I remember, I'll put a link here for you to go and visit that particular lecture. So sodium hydrogen antiport mechanism is an important antiport at the PCT. Okay, it reabsorbs sodium and uses this energy to secrete hydrogen. This hydrogen, once it's in the lumen, guess connects with what? With the incoming filtered bicarbonate. And remember, we are saying it's filtered. It's an important distinction. We're not saying it's new. Okay, so this filtered bicarbonate is coming in. It was minding its own business, but suddenly it was attacked by hydrogen, and hydrogen combined with it and formed, well, you know it now, you know the story, H2CO3, which then dissociated into carbon dioxide and water. And this loop basically is established, and it goes on and on and on at the PCT with the resultant, this bicarbonate, which is basically this bicarbonate. So now you understand. If you stop this bicarbonate coming in, this whole reaction will stop. Agreed? So this bicarbonate is this bicarbonate, and this basically gets filtered, reabsorbed across the basolateral border into the blood. So this way you reabsorb your 99% of the bicarbonate from lumen into blood. Okay, this is one of the main points. Some extra points about this is carbonic anhydrase inhibitors. These are diuretics, a class of diuretics. Again, I'll be recording a dedicated short video on diuretics. But a footnote here on carbonic anhydrase inhibitors, acetazolamide is an example of it. It basically inhibits this transporter, this antiporter, and stops sodium reabsorption. So sodium along with water is just goes through this PCT in the lumen further on and is excreted. So by this, in this way, this drug basically has a diuretic action. Hence, it increases the sodium excretion. Okay, and it's given in certain patients who have ECF overload, a volume overload. Okay, and then there is another thing which I mentioned in the same lecture when we were discussing sodium reabsorption in the sodium handling of the nephron, is volume contraction, is a contraction or alkalosis. So when ECF contracts, what happens? When there is ECF contraction, volume contraction, ECF volume goes down, okay? GFR goes down. When GFR goes down, the filtrate entering the lumen, the amount goes down, the velocity also reduces. When the velocity of this fluid, which is passing through this cell, is reduced, sodium hangs around here a bit more than it usually does. You got that? Decrease ECF volume, decrease GFR, decrease amount of fluid, less in velocity, passing through the PCT in this neighborhood of this antiport mechanism. Sodium hangs around a bit more, stays around a bit more, and this basically increases the reabsorption of sodium because it will facilitate the binding of sodium because it's hanging around here. It's staying around here a bit more because of the decreased velocity. And this reabsorption, since it's coupled with hydrogen secretion, in this scenario, you will end up secreting more hydrogen than you usually do. Okay, and this extra hydrogen will be lost in the urine. So this hydrogen is being lost in the backdrop of ECF volume contraction, i.e., decrease in ECF volume. Here has caused loss of hydrogen. Okay, now if you lose hydrogen, net everything is electroneutral, right? So hydrogen and bicarb are in a balance. Now you have basically tilted that balance, you've disturbed it. You are excreting more hydrogen than you normally do. What will happen in the body fluids? There will be alkalosis. So this is where this term comes up, contraction, alkalosis. Okay, so this is an important point to note, that in ECF volume changes, you have a acid-base disorder. This is actually a very cool question from with a good student in a viva, oral question and answer session. Contraction alkalosis connects the ECF volume concept with an acid-base disorder. And to put a cherry on this complicated cake, I beg your pardon, is if you basically create this alkalosis and the contraction, the decrease in volume is being made by vomiting. Vomiting is interesting in the sense that excessive vomiting basically leads to a loss of volume and what else? A loss of HCl from the stomach, right? So vomiting, unlike dehydration or blood loss or this, that, the other, losing volume by vomiting, excessive vomiting, will not only get rid of volume, ECF volume, but also will create alkalosis by getting rid of the chloride, the acidic ions, right? So this particular metabolic alkalosis is difficult to treat. It's difficult to manage. Why is it difficult to manage? We will talk about this in the third installment of this series of acid-base disorders. But remember that we mentioned it here, because when we go there, I will recall this whole thing and volume contraction over there. Okay. So number two was excretion of hydrogen as a titratable acid. No big issues here. Phosphor buffer system is a very effective buffer system in the sense that it's very efficient in its titration of hydrogen. Where does this whole come from? You remember now this whole thing, it's again the same same reaction going on inside the alpha intercalated cell, which you know is part of DCT. It produces hydrogen, which is not just secreted by dedicated ATP secretors, hydrogen secretors, but also hydrogen potassium antiport mechanism, which I've mentioned in part of potassium homeostasis. The net result is secretion of hydrogen by this cell. This hydrogen is basically titrated against HPO4, which is filtered. So it's being made in the body and it's filtered in the glomerulus and comes to the this part of the tubule, gets titrated with hydrogen, forms H2PO4, which can then be relatively easily excreted. Okay, so this is one part. There are three very important points here. One part is how hydrogen basically titrates against filtered HPO4 to form H2PO4. Another very important point is this bicarbonate. Do you see any bicarbonate here? Nope, there is no bicarbonate here. So how does this bicarbonate come about? Because hydrogen is being used by this reaction. So whenever hydrogen will be titrated against a HPO4 ion and H2PO4 phosphate-based acid is made, a bicarbonate is quote unquote created. We call it new bicarbonate because it just happened to be created by this reaction. Okay. this bicarbonate will be reabsorbed. We call it new bicarbonate, as opposed to, let me just go back, filtered bicarbonate. So at PCT, you have filtered bicarbonate being reabsorbed. Here at the alpha intercalated cell, you have new bicarbonate which is reabsorbed. Very important point to note. Okay, new bicarbonate given in red, so you can't miss it. Okay, the final point about this system is that buffer system, this phosphate buffer system, however, it's very effective. It's a sharp buffer system. But the problem is it's not in the control of kidney. It's not a dedicated acid-base buffer system. It's basically takes its cues from calcium and dietary calcium and bone-related mineral calcium. That is where it basically gets adjusted. It basically gets fluctuated. So mineral metabolism, I beg your pardon, bone metabolism and the kind of diet that you're taking, they will affect how much HPO4 will be filtered at any given time. So the kidney cannot really depend on this sharp buffer system. Although when it's there, it's fantastic, and it's there. A normal basal level is always there, but it cannot be really relied upon when you need a sustained hydrogen bashing buffer system. This is not the choice because, as I mentioned, it's not in. Uh, it's not even made specifically for acid-base disorders. It's something else. It just happens to be such a way chemically that it can titrate hydrogen. Okay, so it's almost incidental, right? Cannot be depended upon. So that's one thing basically. So the number two thing is this hydrogen its titration against phosphate. Finally, we have hydrogen and its and the ammonium form of hydrogen basically, and this is where things, quantitatively speaking, are much better within the control of the kidney. This is a good chunky slide in terms of information. Lots of questions are generated from this particular part of renal function. And two cells are being shown to you. Let's start from these. Proximal, the PCT, and the alpha intercalated cell. Now I'll just explain what's going on. So PCT, this function of PCT has not been discussed up till now. This is brand new information for you. Glutamine is an amino acid. It dissociates by certain enzymes to form ammonia, the gas, and alpha ketoglutarate. Alpha ketoglutarate then forms a new bicarbonate which is reabsorbed. So this is one wing by which you form a new bicarbonate. Each glutamine, when it's broken, gives rise to one new bicarbonate here, and another bicarbonate later on is formed. We will get to that. Okay, so this thing is sorted. Number two, this ammonia, the gas is formed. It's basically combined with internal hydrogen and can be secreted as ammonium ion, number one, or it can be, since it's a gas and it's freely transferable from the cell into the lumen, it basically can take a trip, and it does take a trip through the lumen, through the medullary interstitium, and basically come into the lumen in front of the alpha intercalated cell at the DCT. Just imagine the nephron, a gas is being produced at the PCT. It will percolate through the PCT cells and roam around in the interstitium, and then conveniently gets into the lumen of the DCT. Okay, this is that ammonia which was formed right here. Okay, now this ammonia, very similar to what happened to the HPO4 ion, is basically combined with the secreted hydrogen. This whole thing is exactly the same. This new bicarbonate ion that is made is exactly the same as is made in the phosphate buffer system. However, it is the second new bicarbonate ion in addition to the first new bicarbonate ion that was formed when the original glutamine molecule was broken down. So each glutamine amino acid, when it's broken down, it produces not one, but two bicarbonate, new bicarbonate ions. Okay. Uh, to complete the equation, and ammonia is combined with hydrogen to form ammonium. Ammonium is a weak acid and it's easily excreted. So that's that. Now, one obvious benefit of using this buffer system is that it produces more new bicarbonate ions. So that's one thing. Number two, this whole system basically is tied to acid-base requirements. How is it tied to acid-base requirements? Well, when you have acidosis, guess what happens? When you have acidosis or an acidic change in the ECF fluids, guess what happens to the enzymes which affect glutamine metabolism? You guessed right. They get activated. They get activated by the acidity of the ECF. They get activated and more glutamine than normal starts to be broken down so that more ammonia can be made, more titration can be made, more ammonium can be made, and very crucially, more new bicarbonate can be made. So this is a specific point that I'm raising here, that unlike the phosphate buffer system, the ammonia buffer system has its ties with the acid base world, and it can be relied upon in a wide fluctuation of acid and base pH. Basically, it can be relied upon. It's indigenous to the kidney. It's all of the actors and the reactors are within the kidney. No outside element is controlling it except acid base pH fluctuations, which is welcomed by the kidney. Quantitatively, it produces more new bicarbonate as well. So that's also checked. So this overall is quantitatively brilliant buffer system. Okay. So basically, in acidosis, this, as I mentioned, this whole renal glutamine metabolism gets basically activated. Also, when the urine gets a bit more acidic, it becomes a much more effective trap for ammonium ions. More ammonium ions are trapped in more acidic urine, which clears off the ammonium and hence makes way for this transport to be increased as well. So if the urine here is more acidic, more ammonium gets trapped and cleared and gives the all clear to new ammonium to be secreted. So this whole pathway is facilitated. More ammonium also makes, as I said, more bicarbonate ions, and 50% of hydrogen secretion is through basically this ammonium buffer system, and 50% of your new bicarbonate is made through this buffer system. Okay, so that's one aspect of any change in acid base having its effect on glutamine metabolism and the whole thing that I described. However, if there is chronic acidosis, so the acid-base disorder is not sorted out by all of these mechanisms in the acute timeline, and if it enters into more longer periods, then this ammonium excretion can increase its hydrogen excretion as ammonium ions to 500 milliequivalents a day. So you can imagine that if the person has chronic acidosis, obviously his net acid secretion will be much more, right? So this ammonium buffer system can upgrade itself from having to deal with that 50 to 100 milliequivalent per day in a normal person to 500 milliequivalents a day of hydrogen ions. It can manage it, it can sort it out. So in chronic acidosis, this is the dominant buffer system, both for hydrogen excretion and for new bicarbonate production. Okay, that's that. This is the role of kidney that it plays in acid-base disorders. The final slide would be a comparison between the phosphate buffer system and the ammonium buffer system. We've already discussed this, But let's have three scenarios, and it's nice because it gives you values. So in the normal situation, say the total production of fixed acid is about 50 milliequivalents per day. Out of this 50, so this is nice, it gives you the quantities. You'll remember this better. The phosphate buffer system can get rid of 20, while C, ammonium gets rid of 30. So basically there is a 40-60 ratio, 40% and a 60% ratio between ammonium and phosphate buffer system. Okay. Now, if you increase, there is one scenario where you have increased the production of fixed hydrogen, and there is another where you have decreased the production of or the availability of fixed hydrogen inside the kidney. The increased one is diabetic ketoacidosis. I've explained it. You have increased keto acids in uncontrolled diabetics, and this is the value, say it's 500. The ratio is almost the same. 100 will be titrated against phosphates, and the major bulk of this load will be carried by the ammonium buffer system. In chronic renal failure, why has this total production limited to 50? Is because the GFR has gone down. Okay, the GFR has gone down. There is less production of the filtrate, and hence a less amount of the whole thing is coming through. Um, and even if scrap that, actually, even if you have normal production of you don't have a primarily acid based problem, you only have the problem in the kidney, and you have a normal diet, normal protein diet, you see that basically the excretion of this 50 is divided as 10 and 5. And here you see that the reason for this is basically, hold on. So we see the brunt of the damage to the nephrons is clear here, that the excretion by ammonium buffer system decreases more, because you actually have damaged PCT cells amongst other tubular cells. And hence this system, which used to take the bulk of the responsibility of clearing out extra hydrogen, gets hit and has decreased phenomenally. And the titrable phosphate buffer system also takes a hit, and almost is halved in its titration ability. And as you see that in this scenario, the whole net acid load is not being sorted out. Only about 15 milliequivalents are being barely managed. So that you have net, you retain more acid than you produce, and you have, in chronic renal failure, you have development of metabolic acidosis, because the titration and the excretory mechanisms of the kidney are not working because of damage to the kidney itself. So in chronic renal failure, you will have metabolic acidosis, and therefore it's crucial that these patients are placed on a low protein diet, so that they don't produce even the normal amount of fixed acids that is produced in a normal person, because these people have compromised renal function, and they need to really decrease their protein intake. Okay. Right. So that's that. These are the references of this part of the part one of acid-base balance. Please refer to the two other videos, part two and part three, to have the complete picture, inshallah. Assalamu alaikum wa rahmatullahi wa barakatuh.